This chapter is all about control. So all of that stuff we learned on Tuesday about how microbes grow, we're going to put to use in understanding how to prevent them from growing. And there's really sort of an important historical line in those sort of before aseptic technique and after. Does anybody know what sort of most important discovery or the person who sort of initiated this technique based on what we talked about? Lister, yeah. Which is the origin of the Listerine brand. Yeah. No, and he was really important. So because he was one of many people who contributed to the body of knowledge that led to how we operate around microbes today. But because he was a physician and he was also pretty well read, he sort of synthesized what he was hearing about in the biological world with what he was doing in practice in the field. And he introduced methods that prevent infection wounds that will now, like today, we sort of take for granted. Even if you don't really understand a lot about microbiology, you kind of take for granted that when you go into surgery, they're going to do certain things. Everyone's seen, ER pretty much and know that you scrub your hands and all this stuff. But not very long ago, barely a couple of 100 years ago, this was not even common knowledge. People had no idea. So they did a lot of things that now, if you saw them without being a doctor or an expert, you'd be like horrified that someone was doing that. And how he came up with this was that he had heard about Pasteur's work and he was impressed with it. And he wondered whether the small organisms that were causing food spoilage and other things were also responsible for infections. And so he applied one of the earliest disinfectants, phenol, directly onto damaged tissues. And it actually prevented infection and increased the survival of his patients. He also sort of laid the ground for current methods of sterilizing instruments and maintaining a clean operating environment. That's what we do now. Does anybody know anything about phenol? Would you pour it on yourself, given an opportunity? Why not? It's caustic, yeah. So it was one of the early disinfectants, and we don't use it very frequently because it can cause damage, especially at higher concentrations. But it's better than dying or getting gangrene, right? So it was actually revolutionary. We've come a really long way in a couple of 100 years. So until the late 1800s, even undergoing a minor surgery put you at an extremely high risk of dying, which I think is incredible. And the reason was that people did not understand that you can pass disease by touching things, and they didn't understand especially that there were microbes in the air that could land in an open wound. So now, of course, we understand these things. We use very strict procedures in order to avoid contamination, and most surgeries are relatively safe. Some terminology that we have to know. Sepsis refers to microbial contamination. So asepsis is the absence of significant contamination. That word significant is important. Doesn't mean the absence of any contamination, but we try to minimize it. Aseptic surgical techniques prevent microbial contamination of wounds, and antisepsis is when you remove pathogens from living tissue. Degerming is removing microbes from a limited area. Does anybody have an example of this? Have you ever been to the clinic with your child to get a vaccination, or have you ever taken yourself to get a vaccination? What's the first thing they do? They swab the area with alcohol. That's degerming. So they're just in a small area where you're going to go through the skin, which is your primary protection against disease. Since you're going to penetrate that, you want to degerm the area because harmless organisms on the surface of the skin can actually be harmful if they get into some part of your body that normally they're not found in. And this word biocide or germicide just means the ability to kill microbes. Bacteriostatic agents, on the other hand, do what instead of killing them? They just stop them from growing. So they're still alive, they just can't divide, basically. So there are several levels of control that you have to know in this section. And the definition of sterilization is removal of all microbes. So that would include endospores and viruses. Sterilization does not remove prions. What are prions? Yeah, they're proteins that are infectious. They're not all proteins are like that, but these particular ones are infectious, and they're at the same time, coincidentally, because of their structure, extremely difficult to destroy. When we use the word commercial sterilization, we're mostly focusing on botulism endospores. So you're not so much caring about getting rid of everything, but you're specifically caring about getting rid of botulism endospores because why? Why do we do that? Because it can kill you. That's usually the answer. Disinfection, so different from sterilization, disinfection is a lower level of control where you eliminate most or all pathogens. What does that mean? Are there still things potentially alive? Yes. Okay, so some viable microbes are still there, most likely. And when we use the term disinfectant, we're using it on a surface or an object, an inanimate object. So we'll often refer to these as biocides, germicides, bactericides. Antiseptics are something that we use on living tissue. They could be actually the same chemical, but the context makes a difference in terms of the term that you use. So what are some disinfectants that you may have in your house? Yeah, we've got Clorox bleach, we've got Lysol in the classroom. And antiseptics, for instance, Lister used phenol. There are other ones, right, like iodine and stuff like that. The next sort of level down is decontamination, where you're reducing the levels of pathogens, so just to a point where you can safely handle them. And this would be, for instance, something that's frequently done in food preparation and in situations where you have to share equipment. Sanitization is basically not a specific level of control. It is something that the Food and Safety Administration requires the restaurants and other health or other food service workers to meet. And that's basically reducing microbes to meet the health standards. So it's a somewhat nebulous term. It's going to vary depending on which guidelines you're following. Preservation is when you do something to delay spoilage. And you might, for instance, adjust the conditions. So how would you preserve food if you're going to do it by adjusting conditions?
Add salt.
You could add some salt. So you could add agents that inhibit growth of organisms, so salt and sugar, or you could add chemicals. If you were going to adjust the environmental conditions, what could you do? Say again? Yeah, you could put it in the fridge or in the freezer. Or if it's a salad bar, you could make sure that the ice under the things is not melted. Now, the different kinds of methods of control that you choose are going to really vary depending on the context, right? We're obviously not going to have the same levels of control in a kitchen where you're preparing raw meat as you are at a surgery in a hospital, because it's just not as important, right? So if we're going about our regular lives, we usually just wash things with soap. Like we wash our hands with soap, we clean our dishes with detergent. And soap basically, if you don't already know this, is not really a killing agent. I mean, it can cause damage to microbes because the soap itself can disrupt the plasma membrane, but mostly what it does is just remove stuff. Right? And that's all you're doing when you're washing dishes or washing your hands. You're pretty much just removing the stuff. And what's not affected by that is the normal microbes that live in your body. Okay? And even on your skin, there's lots of microbes in the layers, because we have many layers of skin tissue, right? And so the microbes in there aren't really affected by the soap. You're not washing them off all the time. Now, the exception would be if you add something to your soap, as you might see in the drugstore all over the place, that is a chemical that is bactericidal and that specifically kills, actually is a killing agent, is not just a removal agent. Do you understand the difference? And so if you do that, you could actually start killing some of the microbes that are just harmless or are actually helpful. It is really important to do this if you want to prevent transmission of disease. Why? What are you usually washing off if you're just washing off the surface stuff? Yeah, the thing that, like the cold virus that landed on here, or that kind of, whatever's on your hand when you go to touch a patient. In hospitals, it's especially important to minimize microbes because of the danger of nosocomial infection, which is literally just means a healthcare-associated infection. And most of you are familiar with this term, I imagine, right? Why are most patients more susceptible than the general population? Lowered immune system. They're already challenged. Why are you at a greater risk if you're having an invasive procedure, like a surgery? Right. You're cut through the skin, which is your primary method of defense against disease. And pathogens are most likely to be found in a hospital, more likely than, say, walking around in at least a developed country. Why is that? because patients are there and they're sick, right? So they're actively incubating all these things, and there's lots of body fluids around that might have viruses or bacteria in them. It's really important, obviously, that if we handle patients or we do surgery, we have to sterilize the instrument in order to avoid introducing microbes into the body. And it's important to recognize that it's not just really, those sort of random rare pathogens that you're worried about. There are normal microbes on your body that don't hurt you unless you cut yourself and they get inside, okay? Now, there is this new concern we have about prions because they're very hard to destroy, and so the regular methods of instrument sterilization actually are not that effective against prions, and we have to do extra stuff to prevent them. As we know, in a microbe lab, we take precautions too. In order to protect our samples, usually in this lab especially, because nothing we handle is super dangerous, we're mostly doing that so that our experiments don't go awry, right? But if you're handling something somewhat more dangerous, you also want to protect yourself, and you certainly don't want to be smearing bacteria around and then leaving the site, and then someone else comes along and gets sick. So we use some control methods. We sterilize our media before we use it, right? So those plates that you work with in the lab are already sterilized. And we sterilize stuff after. So once you are done with your experiments today, you're going to put them in the biohazardous waste, and it's going to get autoclaved. We use aseptic technique in the lab to prevent contamination of ourselves and of the sample and of the lab. These are the CDC biosafety levels that you've probably heard. And I'm going to walk you through them and sort of give you an idea so that they make a little more sense. You may already know these, but biosafety level 1 is just microbes that are not known to cause disease. And you can wear gloves and a mask, or you don't necessarily need special precautions, such as non-pathogenic E. coli, right? Because that's something we have in our bodies all the time. It's not really very dangerous. Biosafety level 2 is when we're dealing with something that's either only going to cause a mild disease, or it might cause a more serious disease, but it's non-aerosolized. What does that mean? If it's non-aerosol, what is that referring to? It doesn't travel through the air, okay? So all we have to do is contain it. Since it's not traveling through the air, we just have to physically contain the surfaces that come into contact. The flu is an example. So the flu is an aerosol, but it only gives you the flu. Most people will survive. Lyme disease, dengue fever, which is transmitted by mosquitoes, measles, which is, again, aerosolized, but it usually causes a mild disease. HIV, you might not know this, but it's only a biosafety level 2. Why is that? Is it a mild disease? No, it could be fatal. Most likely without drugs it will be, but why do we put it in this category?
It's not airborne.
Because it's not airborne, right? It's actually quite difficult to transmit. So, and something like MRSA, for instance, also in this level. Now, the next level up, biosafety level 3, is when we're dealing with something that's serious and may be lethal after inhalation. In that case, you just regulate the airflow. So usually you're working in some kind of room that has its own air supply or where there's filters and stuff. Examples of this would be tuberculosis, typhoid fever, anthrax. Biosafety level 4 is the highest level. That's when you're dealing with something that's lethal, aerosolized, and for which there's no cure. In that case, that's when you wear the hazmat suit. So in all of those movies where everyone's wearing the hazmat suit, it's really exaggerating how often you're going to deal with a biosafety level 4. But smallpox would be one example. Hemorrhagic fevers like Ebola would be another example. Now, in food production, we have to keep our surfaces clean and free of microbes, so we do sanitization. And there are a number of ways to do this. It's most common to use heat treatment. It's a very reliable method, but it's not always ideal. Because if you heat treat things like salad, for instance, then obviously you're not going to stay in business very long. Because unless you can get people hooked on wilted salad, this is, you have to find some other way of dealing with it. So radiation can sometimes be used to treat certain foods that don't handle heat very well. Or you can add chemicals to prevent spoilage. And this is regulated by the FDA because adding chemicals can also have detrimental effects, obviously. Now, we have to think about this in water treatment. What we're doing in water treatment is basically trying to make sure there's no pathogens. What does that mean? Is there still living stuff in your water? There likely is still living stuff in your water. And if you live in certain parts of the United States where there's stagnant water and it's hotter, your water might include amoebas that if you shoot them into your nose can eat your brain. So there's definitely stuff in your water. There can be things in your water that if you swallow them are totally harmless because your body will digest them, but if they get into your eyes or other parts of your body, they could actually be harmful. Chlorine is the most used way to disinfect water, but it has some limitations and some concerns because it can actually form some toxic chemicals that are linked to long-term health risks. There are also organisms that are not killed by chlorine. And one example is Cryptosporidium, which causes diarrhea. And so there are regulations in place that require facilities to minimize these toxic byproducts of chlorine and also check for the Cryptosporidium levels in the water. So it's important to recognize that even sort of at the highest level of clean water in the world, there's still stuff in your water. It's not like that water is sterile, for sure. All right, so how do we choose a particular procedure? We have to weigh different things. There's no ideal method that just is best for everything. Everything has a drawback. Everything has things to consider, and there are limitations. We're going to consider simplistically heat, radiation, and chemicals. And how we choose depends on a lot of things, like how many and what kind of microbes are we likely to encounter in that particular context? What are the environmental conditions? What's our risk of infection? And what is the infected item that we're looking at made of? So we're going to start with this. These are physical factors that we need to consider when we're trying to decide how to control growth on an item. So there are some methods that would be inappropriate for certain items. If we're using heat, under what circumstances would that be not an appropriate method to use if we're trying to sterilize an item? Say again? Food, or sometimes food, it's fine to treat it with heat, sometimes not so much. Anything else, something you don't ingest. Think about items that you use in the world or that might be used in a hospital that you cannot heat. Plastics, right? Because a lot of them will melt. Other sensitive items like electronics, for instance, you're not going to put that in an autoclave. So instead you could use radiation, but radiation can damage some plastics. So again, we have to think about that. The most common way that we use to sterilize things in an industrial setting is moist heat or liquid chemicals, right? So autoclaving or certain kinds of liquid chemicals. Under what circumstances would this not be an appropriate method to choose? Again, it's stuff that's going to be moisture sensitive that you don't want to be exposing to autoclaving or to liquid chemicals. So now we want to consider the risk for infection. Medical instruments are categorized according to the risks so that we know how to treat them properly, right? So there's no point in doing the highest level of control that's time-consuming or expensive if it's, a stethoscope. We don't have to put that in the autoclave. It's not like you're going to insert it in someone's body cavity unless you're practicing really weird medicine. So critical items are those that come into contact with body tissues. They must be sterilized, which means what? What's the definition of sterilization? Removing all microbes. Now, what are we potentially not removing is prions, right? So that's, there are new methods to remove prions that are being employed to prevent surgical instruments from contaminating patients with prions. So this would include things that actually are going to penetrate the skin and touch the inside of your body where nobody usually gets in. So that's needles and scalpels. Semi-critical instruments are those that contact mucous membranes. They don't penetrate the body tissue in a way that's sort of really invasive, right? So this could be something like a tube that goes down your nose or some kind of instrument that's going to go into your mouth, other body parts. They must be free of viruses and vegetative bacteria. Endospores are rare and are blocked by mucous membranes, so we don't have to worry about that in this case. That's nice. So things like endoscopes, endotracheal tubes, things that will go, we're touching your mucous membranes, but they're not actually poking into your muscle or through the layers of your skin. Non-critical instruments contact unbroken skin only, so they have a very low risk of transmission. And obviously there are some things we want to, you'll notice that doctors, or if you're handling like stethoscopes and stuff yourself in your job, there are certain precautions that you take because you can still transmit, say, HPV, right, from skin-to-skin contact, but you're not at a high risk of transmission. So the surfaces in a hospital, say, or stethoscopes, blood pressure cuffs, those are non-critical. All right, so now let's consider the biological factors. So these are sort of the physical factors of the thing you're trying to disinfect. Now let's consider the stuff you're trying to kill. Some microbes are harder to kill than others. Okay, so there are some challenges that present it. So if they're heat resistant, that's a challenge. What do we know that is heat resistant? As one example. Say again? Endospores are a great example of being heat resistant, right? The vegetative bacteria can be killed by a regular amount of autoclaving, or pasteurization or some other method, but the endospore can be much more resistant and can still cause a lot of damage. So bacterial endospores, for those you need a higher heat or certain kinds of chemicals. Other things that are heat resistant include protozoans, cysts, and oocysts, and we haven't really talked about this very much. Protozoans are what kind of cell type? They're single-celled, but what kind of cell type? Are they prokaryotic, eukaryotic? They are eukaryotic, and they're animal-like, right? They don't have a cell wall. They can move around. They often are covered in cilia, or they have a eukaryotic flagellum. And again, as I don't want to say vegetative, but as a sort of like in its normal state of living and metabolizing and moving, it's relatively easy to kill. But just like bacteria can make endospores, protozoans can make cysts that are much more resistant. And so they are really resistant to disinfectants and often will be found in fecal matter. If they're human pathogens, that's where you'll find them. They can cause diarrhea if ingested. Mycobacterium species, what's special about these? What do we know about their structure that would make them resistant? The waxy coating, which is made of what? What's it called? I'll give you a hint. Okay, here's my hint. Mycolic acid, yes. Okay, so Mycobacterium species, which include Mycobacterium tuberculosis and Mycobacterium leprae, as well as some nocardial species, which also some of them are pathogens. These guys have that special cell wall structure, and it makes them resistant to chemical treatments, much the way that they're resistant to dyes. There are also some species of the Pseudomonas genus that are resistant to and can actually grow in some disinfectants. Isn't that a kick in the pants? I think that is, just so insulting. And naked viruses, and this is interesting. I think a lot of people, when they think about viral structure, if they do, they think, oh, well, some viruses have an envelope and some don't. So the enveloped viruses are going to be more protected, right? But in actual fact, it makes them in some ways more vulnerable. So the thing about naked viruses is they don't need an envelope to propagate, right? Actually, the envelope isn't there primarily to protect them. The envelope is part of how they get into the cell. And what's it made of? What's the envelope made of? What chemicals is it made of? Where does it come from? The envelope outside of a virus. How do they get it? They get it as they leave the cell and they wrap themselves in the lipid membrane. So it's part of their way of getting in, because they can sneak into the cell that way and then they take it on again as they leave. That lipid membrane is pretty vulnerable, actually, more vulnerable than proteins. So there's certain things that if you damage the envelope, then the virus can't propagate because it can't get into its host. Naked viruses, on the other hand, they don't have an envelope, so they don't care. All right, now let's consider the factor of the number of microbes. It's going to then impact how long you have to heat it or what kinds of chemicals you have to use. And remember also that the death curve of microbes is predictable. Once you know a little bit of information, you can predict a longer term. So if you have some condition, like a certain temperature or a certain percentage of bleach or whatever, some kind of radiation at a certain frequency, then if you know what that does, say, for 10 minutes, you can predict what happens in the next 10 minutes and the 10 minutes after that. Why? Because the death rate is exponential, right? So it's constant. It's like, just like when you're growing, under ideal conditions, the growth rate is exponential. So that means in any particular given amount of time, you're going to double in some period of time, right? Your doubling time might be 20 minutes, it might be 2 hours, but whatever it is, every 20 minutes something happens, or every 2 hours that doubling happens. Similarly, the death curve is exponential. So if you know that 10 minutes of radiation kills some percentage of your population, the next 10 minutes is going to do the same thing. Does that make sense? Okay, so that also means that if you start with a large population, you need more time. Consequently, this is sort of intuitive, right? You just sort of know, well, if I'm trying to kill stuff, then if I wash it off first, then I'm going to, that's sort of step one, and then I can kill it with something. But it's because of this. So if you remove organisms first by washing them, then you do some sterilization, which is why, for instance, when I used to work in decon in the basement of a hospital, the first thing I did, which was so fun, was I would take the instruments and the bedpans and all the stuff that came off the floor and scrub them with the whole suit, very hot. Scrub them, rinse water, so they were like physically to the eye, they were clean. And then you put them in the autoclave. Because what'll happen if you just put all that crap in the autoclave, besides the fact that it's disgusting, if it's still got stuff on it, is all of the body fluids and the stuff that's on there could protect things that are inside, and you just would need a lot more time to kill everything. So, death rate is constant. Here is an example. So let's suppose this is a timed experiment, and you measure deaths every minute, okay? So in the first minute, nobody's dead. In the next minute, there's 900,000 deaths. In the next minute, there's 90,000 deaths, and so on and so on. And this is starting with a million, starting with a count of a million. So you have a million, which would be like, the contents of a colony of bacteria, right? So what is happening is it's not that every minute you're losing a set number of cells, okay? It's not like every minute you lose 10,000 cells. It's that every minute you're losing a certain percentage of the total population. So in the first minute, you're losing 90% of your population, okay? And so 900,000 cells die, leaving 100,000 cells behind. Now, because that's true, if that happens in 1 minute, you know that in the subsequent minute, 90% of the population will die. But you're starting now with a smaller population. Now your population is only 100,000. So 90% of 100,000 is what? 90,000. So if you take 90,000 out of 100,000, you are left with 10,000. Then in the next minute, you're going to lose another 90%. That's what you lose. That's what is remaining. And so on until you get to minute 6 where there's only one guy left. Does that make sense so far? And again, just to remind you, that's because just like growth is exponential in the log phase, death is exponential in the death phase. Or if we're, this is just normal kinetics of growth in a culture that is running out of food, but the same thing applies if we're just using, 10% bleach or whatever wavelength of UV, you're still going to get this log phase of death. And this is just an example to sort of mathematically prove what you intuitively know, which is that if you start with more cells, it takes more time to kill them. If we start out with 1 million cells, then we subject them to the exact same conditions over some period of time. It's going to take, in this example, 6 minutes to kill everything. If we start out with only 1,000 cells, it's only going to take 3 minutes to kill everything. Okay. So now let's think about the environmental conditions that affect what we're going to do. And this comes back to that example I gave you of working in decon, and when the stuff comes off the floor, what's the first thing that I do? I wash everything. What am I removing? I mean, on one level, I'm removing the gross stuff, right? The bodily fluids. But what's in the bodily fluids is potentially dangerous microbes. And also, the bodily fluids themselves would be places where microbes could get food and shelter and that would protect them, especially fecal matter, which has all kinds of stuff that's delicious and a lot of it's sort of waxy. It has this protective quality. There's lots of lipids in it. So any organic material is going to interfere with the ability of heat to get in. And it's going to interfere with the ability of chemicals to get in so that it's going to reduce the effectiveness of whatever you're doing. So you want to remove any dirt, any grease, any bodily fluids before you begin to apply whatever you've decided to do. You want to thoroughly clean it. If you're dealing with something that has a biofilm growing on it, what do you know? Why does that present a challenge? What challenges do biofilms present when we're trying to clean something? Yeah, Biofilms are resistant to chemicals and they're resistant to removal, right? They're strongly attached. They resist chemicals, antibiotics, and removal, and so it's more difficult to deal with. Now, we can also affect how well we clean something, how thoroughly we can sterilize it, by changing the pH and the temperature. Why is it that pH can influence the effectiveness of a particular technique, do you think? What does pH do that we have talked about being concerned about? The wrong pH can do what? Denature proteins. That is going to make a microbe more vulnerable, right? If we're already sort of taking apart its defenses. Here's an example. So household bleach can kill tuberculosis bacteria at 55 degrees in half the time as it does at 50. So increasing the temperature by only 5 degrees greatly increases the effectiveness because we're denaturing proteins. And if we reduce the pH, in addition to applying this heat, it's even more effective because, again, we're hitting those proteins twice as hard. So here are the things that microbial control agents that you would apply, like bleach or certain kinds of detergents. What are their effects? Depending on what you're specifically using, there are several different effects. So they can alter membrane permeability. Again, that plasma membrane, which is important because it's a barrier that keeps stuff outside of the cell out and stuff inside of the cell in, but is also somewhat permeable because we have to have food coming in and waste going out. So if we alter this, then we're going to affect survival, right? Also, we can damage proteins with various agents and we can damage their DNA. Now, can the chemicals we use to kill microbes hurt us? Yes or no?
Depends what you use.
Depends on what you use. Okay, so are we going to be drinking bleach today? Clearly not, okay? So really, whether or not an agent is dangerous for us is really dependent on the kind of effects it's having on microbes and whether our bodies, our cells, would respond in the same way. Many of the microbicides have the potential to hurt us, but not all of them do. Okay, so here are various methods of control that we'll consider. Heat, both moist and dry, filtration, radiation, chemicals, gas, and pressure. And first we'll talk about heat. So there are main advantages of heat is that it's reliable, very predictable, it's safe. Okay, obviously unless you put yourself in the autoclave, it's safe to use. It's fast and it doesn't cost very much. Also, handling heat is not going to hurt you unless you put yourself in the autoclave or stick your hand in the oven. And it can be used to sterilize or to disinfect depending on how long and how high of a heat you use. There are some important terms that we have to throw into this conversation at this time, and you could be tested on them. You may have to define them. You may have to understand them well enough to solve a problem, including them. So thermal death point is the lowest temperature at which all cells in a culture are killed in 10 minutes. Let's take a second to think about that. Thermal death point. Thermal death point is the lowest temperature that will kill all cells in a culture in 10 minutes. Okay, thermal death point is a 10-minute lowest temperature. Now the thermal death time, which sounds really similar, is the time needed to kill all cells at a given temperature. So that could be, we want to know how long is it going to take to kill everything at 55 because the only thing we have is a 55-degree oven, so we have to know how long it's going to take. So we just put them in the 55-degree oven and we see how long it takes, and then that's our thermal death time. And again, this is going to be a minimum time, right? You could put something in the oven at 55 degrees for 2 hours and everything's dead. It's still going to be dead in 3 hours, but we don't care about the 3 hours. We want to know about the 2 hours. Do you understand the distinction? Yeah. Okay. Now, this other term is not heat specific, but I'm going to write the sort of heat specific definition here, because most of the time when we use this term, we are referring to heat. Decimal reduction time is the time needed to kill 90% of cells in a culture at a given temperature. It can also be used to measure the time needed to kill 90% of cells in a culture using whatever condition. But decimal reduction time means how long do you need at whatever condition to kill 90% of the population. So to test whether you understood what I just said, let's go way back in time to this example. What is the decimal reduction time for this particular organism in this particular condition?
1 minute.
1 minute. 1 minute is how long you need in order to kill 90% of the population. Now, what would be our thermal death time? The total time needed to kill everything. 7 minutes, right? Because at 7 minutes, you would predict there'd be 0.1 cells, which is not very much of a cell. You can reasonably assume that that 1 cell is dead. Now, if we were trying to decide what our thermal death point is, we would have to play around with the temperature and find out what temperature would allow the organism to be killed in 10 minutes. And so whatever temperature this is too high, right? It's not the same as the thermal death point. I don't test a lot on the thermal death point because I think it's a confusing concept, but you should know what it is. Mostly, if I have questions on a test, they are going to be about these last two concepts, thermal death time and decimal reduction time, okay? Any questions about these, the distinctions between them? Okay. All right, so again, decimal reduction time. How long does it take to kill 90% of the population in a given condition? All right, so now we're going to talk about moist heat sterilization. Another word for which that many of you will have heard of is what? Steam, more specific, industrial setting, autoclaving. Okay, when we talk about moist heat sterilization, we're usually referring to autoclaving. So it basically denatures proteins irreversibly. That means that if you then take the heat away, the proteins cannot come back from that. They don't refold. Now, one example that's not autoclaving is boiling, right? So that kills most microbes and viruses, doesn't sterilize because endospores can survive. And this is something we do all the time, right? When we make food at home, we're boiling it, or other things, but that's one method that's used. Pasteurization is another form of moist heat sterilization. And autoclaving, a very common one, steam. And in addition to moist heat, it's also, you add this element of pressure. Now, why is pressure helpful? Why does it make the moist heat more effective?
Making the cells more vulnerable.
Yeah, it's making the cells more vulnerable, so it does, the atmospheric pressure is actually going to impact their survival. It also sort of increases the effective temperature because we're adding pressure. Let's talk about pasteurization for a second. So this reduces spoilage organisms and pathogens and was obviously pioneered by Pasteur. Here are some equivalent treatments that all provide pasteurization. So you can do, and this is what Pasteur did. He did a 63-degree treatment for about half an hour. If you don't have a half an hour, because you're working in a factory or whatever, you can do high temperature, short time. So then you just boost your temperature by 9 degrees to 72 degrees, and that only takes then 15 seconds. And this is commonly done for, the things that we're used to using industrially. If we have something that's a little more sensitive, like milk and ice cream, then we do it even higher for a little bit longer. And what is the balance you have to strike here? Why would you have to be careful about how you heat things like milk and ice cream? What's in them? There's a little bit of protein, right? So you can affect the taste if you heat it too long. Now there's also this ultra high temperature you've probably heard of. Have you been to the grocery store and seen UHT stuff? That's that shelf stable milk, right? So instead of being in the dairy case, you can find it on the shelf. It's still milk. It's just been, you've just done, instead of like 15 or 20 seconds of treatment, you do 140 degrees for less than a second. And that basically kills everything. Except those things that can survive. We call them thermoduric, and it's sort of a circular definition because the definition is an organism that can withstand pasteurization. All right, so most of you are familiar with autoclaving. Basically, you have an autoclave chamber. You run steam through it, and then you also subject the contents to high pressure. And basically what the pressure does is, normally if you heat something, there's a point at which the water will boil, right? And then you can't really heat it past that point. But if you add pressure, you can keep everything from going into the vapor phase, and so you can actually go to a higher temperature. Autoclaving is usually around 220 or 120 degrees, so that's higher than boiling. And you're usually doing it at 15 psi's for 15 minutes. This is enough to kill endospores. If you have larger volumes of things, you have to go longer. So for prions, you have to go 11 degrees higher for 4 times the time. And you usually add bleach, which changes the pH. So it's a lot more stringent condition. I'm not going to talk about that in great detail, but I'll come back to it in a second. Now, commercial canning, we use something called a retort. And in commercial canning, what is the thing that we're primarily concerned about? What is the most dangerous thing that can happen if canning goes wrong? What if you're doing canning at home? What should you be worried about? What's the danger of canned goods that are improperly prepared? Right? Clostridium species, these guys that make endospores and can cause botulism. So if you use this particular method, it reduces a huge number of endospores to only 1. And this is like sort of an overkill, right? So it's virtually impossible to have as many endospores as that. So by using this method, hypothetically, if you had more endospores than 10 to the 12 endospores, this method wouldn't work, but that never happens. And we have to do overkill because even just one person dying of food poisoning in industry is considered unacceptable. If we don't do this, endospores can germinate in canned foods. They typically grow in low acid anaerobic conditions, okay? And it's not the bacteria you have to worry about, actually. So when the spore germinates into the bacterium, it's not like the bacterium itself is, living inside or on your cells and causing a problem. It's that it makes a toxin that secretes throughout the whole body, and that is what will kill you. When we make canned foods, we make them so that they're commercially sterilized so that endospores are killed of things like Clostridium botulinum. Now, the endospores of thermophiles may still survive, but those ones are not typically human pathogens. Because remember, what do we know about thermophiles? What are the growth conditions they like? High heat, like way higher than body temperature, like 60 degrees. I mean, most people are not that hot. So it's usually not a concern. All right. Doing high temperature for commercial canning, for instance, is not a universal solution because some foods can't be heated that high. So instead, we could use high pressure. This is one way that we can deal with something like guacamole, which if you pasteurized it would actually be disgusting. So instead, you use pressure. You go really high, actually. 130,000 psi's is a lot of pressure, just in case you're wondering. And what happens when you put that much pressure on an organism is that you actually denature the proteins and you alter the permeability of the plasma membrane. 2 of the 3 things I said could be affected by these different methods. And as a result, even though that's happening to the microbes, the actual product that you're trying to treat is able to maintain its flavor and color. We also frequently use dry heat sterilization. It is less effective than moist heat. And consequently, if you're going to use dry heat, you have to go longer and you have to use a higher temperature. Now, one of the most effective things to do is kill something by oxidation, which totally denatures proteins. And what do we do in the lab all the time that is a fairly effective form of oxidation? Yeah, we put things into the fire, right? This is also why when you're heat fixing, what do you not do? You don't flip it over so your sample's next to the flame because you're going to then actually burn your sample. If we're not able to flame something, because that wouldn't be so good for it, we can do a hot air oven. If we are trying to get rid of something completely, we use an incinerator, and it oxidizes cells down to their ashes and is used to destroy medical waste and also carcasses. So this is just a reminder of sort of the difference between using moist heat and dry heat. So here's a moist heat with pressure autoclaving. Normally you do this for 120, 121 degrees for 15 minutes. If you want to get the equivalent amount of sterilization from a hot air oven, you have to go 50 degrees more, so 170 degrees for 2 hours. And it is important that you know this for a test. You should just memorize that to the best of your ability. Okay, let's talk about filtration. What is that? Have any of you ever done filter sterilizing in your job or in your workplace? So this is for those things that can't be heat treated, and typically they're things that are liquids. So what you do is you have your liquid that you need to treat in a container, and you pour it through a filter. And the filter has little holes in it. The holes have a known size, and depending on the size, we label the filters that way. So typically we use a small pore size, which is 0.2 microns. How many nanometers is that? 0.2 microns. How many nanometers are there in 1 micron? 1,000. Okay, so 0.2 microns is how many nanometers? What's 0.2 times 1,000? Who's good at math today? 200, yes. So 200 nanometers, 0.2 microns. Why is this a good size if we're trying to filter out microbes that can make you sick? What is it going to filter out? Let's see. How big are eukaryotic cells? They're bigger than that for sure. They're usually like 30 to 50 microns, so they're definitely going to be filtered out. What about bacteria? How big are they? How many microns-ish? Like 1 to 5. So they're going to be filtered out? Most of them. Now there are some that are really tiny. They're outside, they're much smaller than usual. Those would get through. But most of them, most bacteria are going to be filtered out by this. Certainly protozoans will be filtered out. What isn't going to be filtered out probably? Viruses. Yeah. So unless they're very large. One example of a large virus that would be filtered out is Ebola, because it's about 700 to 800 nanometers. So, but you can use this to filter most of the microbes you're likely to encounter. You can also filter air with HEPA filters that removes nearly all microbes from air, which is similar pore size. So here's just an example of what that looks like. You have this sterile collection unit. You have the filter. You pour this into the top part. It goes through the filter. You have some kind of vacuum that's sucking air through so that it'll come through. You don't just rely on gravity. Membrane filters typically are 0.2 micron filters that will remove bacteria, and HEPA filters are roughly 0.3 microns. Okay, so another method we can use is radiation. There's lots of forms of radiation, some of which are not particularly harmful, such as what? What are we exposed to all day long that really isn't harmful to us that is still considered radiation? The sunlight, so visible light, okay? So here is the radiation spectrum from really, really small wavelengths to really, really long wavelengths. Like this is a kilometer long. Radio waves can be really long. Gamma rays are really, really short. So we start with the shortest, which is gamma rays. X-rays are pretty short. UV, not as short. Then we have visible light, which is in the 100 nanometer range, right? So visible light goes from about 400 to 750 nanometers in the size of the wavelength. So, this is not particularly harmful. Ultraviolet light, is that harmful? We do have it in our sunlight. What do we know about UV that is potentially dangerous? It can be associated with skin cancer. Do we know why? Why UV does have that effect? Yes. right, if the DNA can penetrate to the cell nucleus, then it can actually chop the DNA. Now, because this is such a common thing, most cells on the Earth have the ability to repair UV-induced damage. So it either can chop the DNA, or sometimes what it'll do is the base pairs that are next to each other, if they happen to be thymines, they can be stuck together, making it really hard to read for all the enzymes that have to read the DNA. Because this is the case, we have enzymes in our cells that will detect that kind of damage and fix it, but they're not very good at getting the fix right. So they're just like, emergency, have to fix the DNA, but they might fix it wrong and introduce typos into your DNA, which are mutations. All right, so X-rays are more damaging than UV, and gamma rays are the worst. And that is because as the wavelength of the radiation gets smaller, it has much higher energy, so it's going to hit you harder. So these guys, gamma rays and X-rays, are what we call ionizing radiation, and these are the kinds of things that we think of typically when we think about a nuclear accident, for instance. They cause ions to come off of the atoms that they hit, and those, the ionizing radiation causes a lot more damage. So gamma rays and X-rays can actually just destroy DNA. They actually damage membranes. They can react with oxygen, making reactive oxygen species. Remember, oxygen can get electrons, and then it's really reactive and can run around damaging things. It can even ionize water, making it a dangerous form of the hydroxyl ion. Where do we use it? We can use it to sterilize heat-sensitive materials because, you know, just like it's going to damage us, it's going to also damage prokaryotic cells and other microbes. It's been approved to treat a lot of foods that are heat-sensitive, and also to kill parasites that might be hard to locate. Certain kinds of meat can be radiated. Now, the wavelength of UV that's typically used is 260 nanometers. UV varies in its length. It's a spectrum that runs from 100 to 400, so it's just below visible light. So between 100 and 400 is considered UV. And the different wavelengths have different effects. 260 is the wavelength that is able to damage DNA. So that's going to be the most effective if we're trying to kill bacteria, for instance. Obviously, also, if we're using that, we don't want to be shining it on ourselves, right? Because we can still affect their own DNA. So the uses and drawbacks, it's used to destroy microbes in air. Have you seen those UV air things? In my building, we have some near the top of the entrances and exits, and it UVs the light, or it UVs the air as it comes in and out of the building. Also, have you ever seen a UV sterilizing method for water? Some of the new water canteens have UV in them. So what they do is instead of having bottled water trucked in, they have the water coming in from a regular water source, and they just UV the water as it comes through the tap. Now, the limitation of UV is that its penetrating power is very low. So this is why if you're driven across the country and if you drop the window, stick your arm out of it, then this arm gets sunburned and the rest of you is fine. Even though, you have obviously exposure from all of the windows, it's because the glass is actually enough to deflect most UV. So if there's glass or a piece of plastic in the way, then the UV can't get in very well. Obviously, you have to use UV carefully. We're going to use UV in the lab in this class, and when we do that, we will take precautions not to hurt ourselves with it. Microwaves? Anyone use a microwave? Yes? Okay. Well, so we have microwaves. Microwaves are on the other side of visible light, so they're much longer. And what do you think? Are they going to be more or less dangerous than something like X-rays or UV? Less, right? Because they have a lot less energy. What's dangerous about them? Would you stick yourself in the microwave? What do microwaves do? Primarily, what do they affect? They make water hot, which we're made of. And if we are microwaved, what's happening to our water is getting hotter, and so consequently, what's happening? Basically, our proteins are all falling apart. So they have less energy. They can destroy indirectly, but it's indirect, right? So the microwaves themselves don't damage us or organisms. It's the fact that they heat water, and there's a certain point at which that's not okay. All right, so that's why you don't want to stick yourself or your cat or anything else in the microwave. However, because it's an indirect effect, and have you ever noticed that like when you put something in the microwave, you know, parts of that substance are really hot and parts of it not so much? So what does that suggest about the usefulness of this as a sterilization technique? You can't rely on it. It's hard to sort of predictably, evenly get everything. And it's basically only as effective as the temperature that you can reach for a certain period of time. So something like boiling or putting in the oven is going to be way easier to control. So a lot of times, if you microwave something and there's something bad in there, it's still alive. Okay, we are going to talk about chemicals as a method of disinfection. And there's some things we have to consider. One of them is toxicity. So if we're using a chemical, we have to sort of think about how effective is it going to be versus how dangerous is it for us. Also, we want to think about whether or not it's effective if there's organic material around. How compatible is it with the material being treated? Meaning, is it going to damage what we're trying to clean? And how much does it cost to use? There are a variety of classes of chemicals. And I'm going to just see where we are. Okay. Yeah. Very close to our break. I just wanted to see where we are so you all can just hang in there for just a few more minutes before we take a break. All right, so there are sterilants, which do what? We're trying to sterilize things so they would destroy all microbes. We would use these for heat-sensitive critical instruments. Because, of course, we could sterilize instruments using heat, right? Autoclaving or a hot air oven. But there might be things like plastics we can't do that too, so we could use a chemical instead. High-level disinfectants would be things that destroy viruses and vegetative cells but don't kill endospores. So then we would use something like a semi-critical instrument. Because why? Semi-critical instruments are ones that do what? They're not the needles and scalpels. They aren't going through your skin. What are they doing? Semi-critical instruments. They're in contact with what? They're not penetrating your skin. They are going in your body somehow. Where are they going? In through your mucous membranes, right? And the reason we don't have to worry about endospores is because we're naturally able to handle endospores. If they're in touch with our mucous membranes, they can't get into the body. Intermediate level disinfectants, less effective obviously, destroy vegetative bacteria, fungi, most viruses, not all. And we would use them for a non-critical instrument, such as a stethoscope, for instance. Low-level disinfectants destroy fungi, vegetative bacteria, except not mycobacteria, and they also destroy enveloped viruses, but probably not naked viruses. And this is something we would use to disinfect, you know, the furniture, the floor, and the walls. All right, so what we're going to talk about after the break is how do we know if what we're doing is working. We'll come back in about 5 minutes and talk about that.